Okay, welcome everyone to our next talk this morning, this on the hour. The topic of the talk is machine learning for chronic disease management, and we have Professor Rahul Krishnan to present the talk this hour. A little bit of bio about Professor Krishnan. He's an assistant professor of computer science and medicine. He's also a member of the Vector Institute, where he holds a CIFAR AI Chair. His research has developed new algorithms for probabilistic inference and applied machine learning to problems in healthcare, such as modeling disease progression and risk stratification. He received his MS from New York University and his Ph.D. in computer science from MIT in 2020. And we're going to listen to Professor Krishnan soon. Just a reminder, if you have any question during the talk, just put that in the chat. We'll call up the time for questions to relay that to Professor Krishnan. Just put it in the chat. You don't need to do much about that. So, and then also encourage our participants to visit the booth and take advantage of the wonderful prizes from our sponsors too. Yeah. If you missed any talk, you can click on the replay button to go back to the talk that you missed. So that's the brief announcement. So Professor Krishnan, we have attention for the next 45 minutes.
Thanks so much, Franklin. Hi, everyone. My name is Rahul. I'm an assistant professor in CS and LMP at the University of Toronto, and I'm going to tell you today about machine learning for chronic disease management. So I should start out with a disclaimer that I'm going to talk to you about ideas that lie at the intersection of healthcare and medicine, but the contents of this talk should not be interpreted in any way as medical advice. So let's jump into it. If we think about chronic diseases among Canadian adults, there's a surprising statistic that's been released by the Public Health Canada, which is that about 44% of adults over the age of 20 have at least one of 10 common chronic conditions. And these could range from anything from things like hypertension to osteoarthritis, diabetes, COPD, or even cancers. And so the economic burden in the context of a publicly funded healthcare system like Canada is one that's gradually rising over time. And so this begs the obvious question, which is, why should we use data to think about finding solutions to clinical problems that might arise in the context of chronic diseases and chronic conditions? And so cost is one reason, but at the end of the day, what we really should strive for is thinking about how to make use of data to make patients' lives better, to understand diseases better, and perhaps to help clinicians develop new treatment protocols that would decrease the effect that these diseases have on the underlying population. And so the scientific outcome of this kind of research would be to understand disease better, and a potential clinical outcome here would be to make the practice of healthcare more efficient by building software tools whose insights are driven by previous data that exists in the context of chronic diseases. And so the outline for this talk today is really going to tell you about two areas. So this talk is split up into two subgroups. In the first act, we'll talk about modeling the progression of disease biomarkers in the context of disease progression. And in the second act, we'll talk about self-supervised learning for histopathological data. And really, I'm going to focus today on some of the research advances. But what I'll do as we go along is give you why these advances in research can help drive some of the new software that could exist in 10 or even 5 or 6 years into the future. So let's jump into it. What we've seen over the last several decades or so is an exponential rise in the amount of healthcare data that's being collected about an individual. Now, this could range from anything like medical images, x-rays, radiographs, inpatient health monitors for individuals who've been admitted into the hospital, clinical examinations. So when the doctor sees a patient, he or she writes down a note that describes the characteristics that the patient has, what their past treatment history was, patient-generated health data. Everything from your Apple Watch that you might be wearing today to your phone is tracking information about you that has proxies of your underlying health condition. And finally, lab results for which, for example, standard blood panels. And the combination of these myriad of health data, when combined together, can, for example, be used to derive new and interesting insights about diseases that weren't evidently obvious from looking at patient experiences in the context of a single doctor. So essentially, these large collections of data stores aggregate experiences that doctors have over millions and millions of patients, and from that, we can derive new insights about subtypes that diseases might have, or even use it to drive software to help doctors make their daily lives more efficient. The challenge, however, is that clinical data is quite complex. And if we were to take a bird's eye view of what clinical data looks like, it might have a multi-scale structure. So at the lower scale, we would have information on patient genetics. At a scale above, we would have things like imaging and lab tests that give us a view into the functioning, healthy or otherwise, of the organ systems of the patient. At a view above that, we would have subjective clinical notes that to a large degree characterize clinical judgment about the patient. And finally, we would have population level statistics that characterize disease incidence within an overall population. And these different scales span different scales of the human body, but more importantly, these data aren't static. They often change over time as the disease either gets more or less severe in the context of a single patient. And so this begs the obvious question, which is how can we build models to capture this degree of complexity in patient data? You have a variety of different modalities like X-rays, lab tests, and genetics. Each of them can be very, very high dimensional. So how do we leverage the tools that have been developed in the machine learning community to help build software for clinicians that can adequately tackle some of the challenges that arise as a consequence of thinking about how various scales of the disease behave within the body? And so this talk is going to focus on the modality of imaging as well as lab tests, but the broader research agenda, and if you're thinking hard about how to make chronic disease management more effective, it's important to think about the bigger picture as well. So one of the popular classes of machine learning algorithms that's seen a lot of success in recent years are deep neural networks. And in the context of images, these are essentially compositional parametric functions that if you apply them to an image, automatically learn how to extract the right features for a particular predictive task at hand. And so this is an example, say, of a 3 or 4 layer convolutional neural network that automatically learns to extract high and low level features of this image of 2 boats on the left-hand side and turns that into representations, which are effectively just numbers that characterize the likelihood, for example, of a boat being in the picture. And from these representations, we can, and this is depicted on the right-hand side of this image, just train a linear classifier on top of this representation to predict the outcome of interest. And so what we found in the past few years is that the last few hidden layers in a trained deep neural network can often be reused for different tasks. So the pipeline of machine learning, which many of you may be quite familiar with, is as follows. You collect a large data set, you perform some compute on that data set, and often that compute corresponds to training a machine learning model using the data. And think of this computation as effectively the accurate and useful extraction of patterns that are common across the entire data set and incorporating those patterns into this predictive model. And finally, you would use that model as a proxy for all of the patterns that you've extracted from the data and deploy it in the field on data that it hasn't seen before. And so the question that we're going to ask is, what can machine learning do for cancer care? And I've told you that healthcare data is in particular very, very challenging to deal with because of the high dimensionality, the multimodality of the data. And I've told you that deep neural networks, at least in the context of images, have made a large impact. And so in this talk, what I'm hoping to give you is an idea that the same advances that have been applied in the context of images can have utility in the context of medical images and medical clinical data as well. But it's not enough to just apply the method as it would be in the context of computer vision, because computer vision is a field where labels are plentiful. Medical images and medicine in general is a field where labels are not plentiful. It's difficult to find time for doctors to annotate images, and it's difficult to get large, accurately labeled data sets of image data and laboratory data. And so we have to think a little out of the box if we want to make machine learning practical for healthcare. So let's jump in. And so the first part that I'm going to tell you about today is modeling the progression of disease biomarkers. And so this is work that's been done in collaboration with Zishan Hussain, an MD Ph.D. candidate at MIT. And this was work published this year at ICML. So if we take a really oversimplified view of chronic disease care, and in this case, we're going to focus in on cancer care, we can think of what's happening inside the human body as something like this picture. We have some notion of time on the x-axis, and on the y-axis, we have some notion of disease burden. And so disease burden is a latent quantity that is never truly observed in the data itself, but it is a hypothesis that we believe that characterizes how severe the disease is within the patient. And so, you have the disease burden rising initially up until the point of diagnosis, at which point you have the doctors applying some form of intervention. So this could be first-line therapy if this was a cancer. And it works for a while, but eventually it fails, leading to relapse. And the doctor tries both second and third-line therapy, which ultimately fail in the context of this patient. This view has some interesting and useful ideas that we can leverage in the context of thinking about new software tools for chronic disease management. And so in a conversation with a collaborator in discussing the view that I showed you in the previous slide, the collaborator mentioned that, well, it might be interesting to know what the trajectory of a patient looks like early on, right after diagnosis. And so the reason why it's interesting is because it might inform the choice of whether or not to provide aggressive treatment. And so in this case, we have treatment A and treatment B. We have that are two options that the clinician is considering. And if we had a model that could forecast what the patient's disease burden looks like under these two different choices of treatment, it might provide the doctor, and the model is accurate, a suggestion that perhaps in this case, the combination therapy of VRD is a more accurate, is a better choice at this point in time. And so what we set out to do in the context of this research project is to think hard about how can we build good tools to forecast clinical biomarkers in the context of patients suffering from cancer. So this notion of modeling clinical biomarkers often goes under the names of disease progression. And so we're modeling the disease progression using electronic healthcare records. So I'm not sure how many of you know what an electronic healthcare record looks like, but I'm going to present this cartoon picture to you to give you a high-level sense of what that is. So what you have on the left-hand side are a set of baseline statistics. And so these could be the gender of the patient, some basic information about genetics, which is often collected in the context of cancer care. And these are essentially static covariates that don't vary with time. On the right-hand side are the time-varying or longitudinal covariates, and these include things like treatment. So U1 and U2 denote the random variables associated with the multiple treatments that the patient might be prescribed over time. And so this cartoon says there's only 2 treatments, and the indication of a dot indicates that the treatment was prescribed. In addition to the treatments, we also often have data on biomarkers. So these could be the different laboratory values that are often used as a proxy by the doctor and the clinical system in general for how severe the disease is within the patient. So remember, I told you that there's some notion of a disease burden, but disease burden is never directly observed. It is only observed through the lens of these various lab values that the doctor actually can run a test on for the patient to approximate. And so the task from a machine learning perspective for a single patient eye is one of unsupervised learning. So what we want to do here in the context of modeling disease progression with EHRs is unsupervised modeling of clinical biomarkers, conditional on a patient's treatment regimen, as well as their baseline data. And so we want to maximize the probability under some given model of Xi, which are the time-varying biomarkers, given that we know their treatment policy and their baseline statistics. So in the context of images, each random variable, each observation, X, could be a vector where a number in that vector represents the intensity of a pixel. In healthcare, these vectors often represent different features that are relevant for the task at hand. And so if we wanted to model a cancer like multiple myeloma, which is the application of interest here, then we would have, for example, in the first dimension of the vector, the serum IgG levels of the patient. the third dimension might have the serum IgA, and the last dimension might have the serum M protein. And so we essentially want to model how these numbers vary across time. And in this cartoon depiction, there seems to be no variance at all. So what are the current approaches that have been used? Well, latent variable models have a long history of use in the context of disease progression. And so a example of a latent variable model might be something like a hidden Markov model, or in this case, on the right-hand side, a Gaussian state space model, where the key difference between a hidden Markov model and a Gaussian state space model is whether or not the latent process that is used to model the evolution of the time-varying clinical biomarkers is discrete or continuous. So this time-varying latent process is essentially a proxy for the unknown disease burden of the patient. And we'll focus in on the case where these Z's, these latent variables, are going to be continuous valued. And so this is, again, noting that what we might expect is to have the model learn the latent disease burden and incorporate that information into the latent variables of the statistical model that we posit. So in the context of traditional approaches, there are two important limitations. The first one is that a lot of models in the past have been linear. And so you could have a linear state space model where each of the functional representations in these arrows that you see on the diagram on the right-hand side are linear functions. And this linearity can be a limitation because often we see that the progression of clinical biomarkers is heavily nonlinear. The problem is that if we were to just take the current set of tools for nonlinear sequential modeling from deep learning and apply them as is to this problem, we'd often run into overfitting. And so extending to nonlinear state space models often suffers from overfitting because in practice, in clinical data, we don't have millions and millions of training examples. We might have a few hundred or a few thousand training examples. And so our approach was really to think hard about how can we do good unsupervised learning for time-varying sequential clinical biomarkers in the context of patient data when data is scarce. And when data is scarce, we have to trade off between bias and variance. And so our idea is to think of how to encode inductive biases into the neural architectures that are used in the transition function, which is this function that decides how the latent disease burden of this patient evolves over time, and learn this new model. And so there's two key ideas that we incorporated in our work. One was to explicitly model that a patient undergoes discrete lines of therapy. And the second was to think about how to approximate the mechanism of drug effect. I'll focus in on the second aspect of it, which was essentially the idea of developing new neural architectures that have been inspired from the pharmacology community. And so the pharmacology community, the PK-PD, and the PK-PD community has spent a lot of time thinking about how to build accurate dynamics of cancer biomarkers in and the effect that a single drug has on them. And so there's been classical models that have been studied in this literature, such as the log cell kill model, that control how tumor volume relates to the intensity of anti-cancer medication that's been prescribed. Our goal here is to take those traditional PK-PD models, which typically work in the context of a single intervention and a single biomarker, and extend them to the case where we're interested in the effect of multiple biomarkers and the effect that they have on the latent disease burden. This is an example of one such treatment effect function that we derive in this paper, which ends up capturing the idea that after any given treatment, a patient is likely to end up having a slow, gradual relapse after the initial point. And so these different curves in the plot on the left-hand side describe the different patterns that this mathematical equation, and this is the most mathy slide in this talk so far, actually represent. What we did in our work is to create a neural architecture that captures three different distinct PK-PD mechanisms. And we use another idea from deep learning, namely neural attention, to characterize the differential effect that these different PK-PD mechanisms could have on the latent disease burden. And we call this model we're going to denote this model the SSM PK-PD. And so one obvious question at this point is, you've told me that there are existing models that work, such as a linear model, a linear state space model, a nonlinear state space model, and we could also have something like a recurrent neural network or a GRU. And you've told me that these, on the one end of the spectrum, a linear model might underfit, and on the other end of the spectrum, a nonlinear model might overfit. And so you see that play out in the equations herein, where if you look at the number associated with the test time negative log likelihood, you see that it's higher than the SSM PK-PD, denoting underfitting. Note that the nonlinear models also remain higher than the SSM PK-PD, but for a different reason. It's due to overfitting. And so the SSM PK-PD seems to hit the sweet spot between underfitting and overfitting by incorporating the use of domain knowledge into the neural architecture that's used for the state space model. And in addition, once we have this model that we've learned, we can use it for the task that I motivated at the beginning of this aim, which is namely patient forecasting. We can look at how well the model does, in this case, relative to a linear model, when trying to forecast a set of clinical biomarkers, serum IgG and serum lambda, for a single patient. So the experiment that we did here is that we took all of the patient's data up to time point 10, so that's about 20 months, and we asked the model to forecast how it thinks this biomarker would evolve in the future, conditional on having seen the first 20 months of patient data. And you can see here that the black curve in general has a higher fidelity fit to the future outcomes of this patient than the orange curve. At least Act 1, what we've done in this work is that we've developed a new attention-based neural architecture inspired by pharmacology that captures some of the responses that combinations of drugs can have in representation or latent space. And we found that the use of this neural architecture allows nonlinear state space models to generalize well, even when data is scarce. I'd say outright that the context of this experiments were perhaps done on a single clinical cohort of patients suffering from multiple myeloma. And this is data that's been supported and provided by the Multiple Myeloma Research Foundation and the COMPASS study. What we need in order to make sure that this method actually works on a much broader cohort. And so one of the key limitations of this work is that it does need further validation on a much, much larger real-world cohort. So in addition to evaluating and validating our results on a larger cohort, there's some really interesting future work that comes about as a consequence of being able to do good forecasting. And one of them is being able to ask counterfactuals, which is the question that our clinical collaborator asked and motivated some of this work at the very beginning, which is, can you tell me what will happen under two different treatment protocols that I might want to start the patient on at the very onset of their diagnosis? And some of the key challenges here are the fact that treatment assignment is not always random, which means learning these models from registry data do not give you a model that is capable of answering these counterfactuals. This is also true for a technical assumption known as positivity. And in reality, we need new technical and data aggregation solutions to tackle some of these really thorny issues that arise when trying to do causal and counterfactual inference from observational data, while continuing to use flexible model families to learn the patterns by which clinical covariates change over time with. And so with that, I'm going to jump into the second act of the talk. So now the second act of the talk is about a slightly different kind of data modality, namely histopathological image data. And so this work has been done in collaboration with Richard Chen, who's a third-year Ph.D. student at Harvard University when he was an intern with me. So I'm not sure how many of you are familiar with what the workflow in histopathology looks like, but it's essentially, at a very high level, the microscopic examination of tissue samples to study diseases and their different presentation. What does that mean? When the doctor has a suspicion that a patient has cancer or has a confirmed diagnosis of cancer, particularly in the context of solid cancers, there's a protocol that is put into place. So a surgery, a biopsy, or in patients who have already died, an autopsy is done for excision of this tissue. The tissue is placed in a fixative to stabilize it and to prevent decomposition. And finally, this tissue is investigated under a microscope. So this is the pipeline that has existed for many decades now, and histopathological images are routinely used for clinical diagnoses of cancer. Now, I've already told you that deep learning in general seems to be a very good fit for image data. And so the key question here is, how can we use some of the advances in machine learning to automate the process of decision-making or build tools to aid decision-making from histopathological image data? So currently, what we have is the following pipeline, where we have two broad classes of approaches that have been taken in order to characterize and build predictive models from histopathological data. One of the key challenges I should highlight about histopathological data that makes it very different from image data as is the fact that histopathological slides are much bigger in size and in the number of pixels than say data coming from ImageNet. So ImageNet is maybe 256 by 256 pixels. Each histopathological image at 20x resolution is 200,000 by 300,000 pixels. So that is gigabytes of gigabytes of data for a single image. And the enormous size that histopathological images have translate into a variety of very technically challenging engineering problems that arise even from trying to apply simple machine learning models to this kind of data. And so in the bottom row, what you see is an example of multiple instance learning, where a set of, where what's done is that the histopathological slide is cut up into a set of patches, and each of those patches is fed into an encoder, which is typically a ResNet encoder, and the entire image is treated as a bag of patches, where each patch level representation comes about as a consequence of applying this encoder to each of the patch. And finally, multiple instance learning is done in order to predict whether or not, for example, the tissue sample is cancerous or benign. And multiple instance learning typically involves aggregating these bags of patches by averaging them, taking the max over them, or even learning a model over them to make a single representation of this whole slide image. So that's the multiple instance learning view of how to do machine learning with computational histopathology. There's the graph convolutional neural network view, which says that we should think about building graph neural networks that connect the different nuclei within a histopathological image in a fine-grained graph. And there have been models applied to try and do this automatically as well. And so one of the important aspects that's already been shown in the literature is the fact that these methods seem to do a really good job at capturing pathological variants in histopathological images. And so I'll focus in on the bottom row here. If you have the whole slide H&E image on the left side, we can use the attention maps from some of the models that I described in the previous slide to look at what is relevant for a diagnosis of benign or cancerous. And we see that the attention maps have a high degree of concordance with genetic markers, and this kind of experiment has been studied in a few different contexts. And so attention weights seem to saliently localize these tumor regions in binary classification tasks of benign and metastasis. But one of the biggest limitations of the current paradigm is the fact that it requires a large degree of manual annotation. And so it requires clinical domain knowledge to label these image regions in a whole slide image with known morphological phenotypes. And so these would form patch level tasks. And if you wanted to make predictions or prognostic decisions from the entire whole slide image, you would need subjective slide level labels of the entire whole slide image. And so the question that we began asking is, how can we identify new phenotypic biomarkers? And what are we missing in current decision-making that can guide prognosis? And so one of the key challenges that we decided to tackle here was the fact that in the status quo, the current pipelines for creating representations of whole site images often make use of the ResNet 50 architectures that have been pre-trained on ImageNet. And what we wanted to do is ask, can we do better than that? So the class of methods that we use in our work builds upon self-supervised learning. And so self-supervised learning, and unfortunately I don't have time to go into the technical details of it, is a class of methods in deep learning that trades off the use of domain knowledge in order to learn representations with labels. So instead of saying, I want to learn a representation that can predict a boat really well, you try and learn a representation that is accurately able to distinguish images that are similar to this image, or patches that come from the same image relative to patches that do not come from the same image. And so by incorporating ideas of what we would like our representation to be robust to, it turns out that we learn good representations through that process. And that broad class of methods is often known as self-supervised learning. And it does not need any pixel level annotation in order to learn good representations. And so we build upon lines of work on self-supervised learning for histopathological images. So specifically, what's different in what we do is to think about how we can use some of the latest advances in self-supervised learning in order to do good representation learning for computational histopathology. So we use a method known as DINO, which is a method for self-supervised learning that uses two different networks and compares whether or not their predictions are similar or different in order to guide the estimation of representations. So the key takeaway here from this slide is that we have a method that takes in a histological H&E image patch, and by applying this method DINO at the end of our training procedure, we end up with a representation that is informative about some of the underlying aspects of histopathological images. So how can we test that? So to test that, we did the following study design. We took all of the available histopathological images that we had in the context of the TCGA lung cohort. So we had a two-step procedure. We used self-supervised learning to learn representations of whole site images. And in the second step, we evaluated the quality of these representations on a variety of tasks of interest. So we tried predicting organ-specific and versus pan-cancer training on TCGA lung. And we compared our results with several state-of-the-art methods, including SimClear and SimSciM, which are different kinds of methods for self-supervised learning. And the slide level tasks that we evaluated all of these representations on included characterizing the difference between adenocarcinomas versus small cell lung cancer, thinking about how to do good survival analysis, as well as mutation prediction. So I'm going to present, and they are a small vignette of the broader set of results that we have that's currently in a paper in submission. And so on the left-hand side, on the top left, are examples of lung adenocarcinomas, and on the right-hand side are examples of squamous cell carcinomas. And I put these images there for you to try and give you a sense of the relative visual difficulty of distinguishing between the two for the human eye. And just to say that in both cases, we see these long strips of these tissue samples, making it not very clear or obvious to at least an untrained viewer why these two are different. And so at the bottom row, what we see here are the evaluations and the accuracies of a variety of different methods that we've tested on this task of distinguishing between adenocarcinomas versus squamous cell carcinomas. And so the key takeaway here is that what we find is that DINO with vision transformers seems to do really, really well in terms of raw accuracy numbers. But at 100%, it outperforms the other methods. But the most interesting result that we found is that it was able to outperform the other numbers using only 25% of the labels in order to fine-tune the self-supervised representation. Put differently, what this is suggesting is that self-supervised feature extractors from DINO tend to be more data efficient than the naive use of a pre-trained ResNet on ImageNet for this task of subtyping. And another really interesting result that we found is that after some work on thinking of how to introspect what the model is looking at, we can create these artificial attention maps that look at different attention heads in the architecture of our model. And what we find is that different attention heads seem to disentangle concepts in this image on the left-hand side. So if you think as a viewer of what really composes the H&E image on the left-hand side, you'll see that it's made of cells. These cells might have different types. We see that there are spatial locations associated with the cell, and we see that there's spaces in between those cells. And it turns out that the same decomposition that I've described to you in words is what the model is learning automatically when it does self-supervised learning. And so the three plots in the middle showcase what the model automatically learns with no annotations whatsoever. And the plot on the right is an example of running a segmentation model on the original H&E image, showcasing that self-supervised learning attempts to learn these disentangled representations of histopathological data that have a high degree of concordance with human annotatable labels of interest. And so there's two important implications. And so the first one really is that data efficiency enables application of this kind of technique to decision-making with rare cancers, for which we might have 5, 10, or only 20 patients in a hospital coming in every 5 or 6 years. Secondly, the aspect of this model that it learns disentangled representations is useful because we can think of how to use the attention weights as proxies for new and different phenotypes and discover how they relate to outcomes of interest. And so I'm not going to say that this work is complete. There's always more research to do. We want to understand why the representations of self-supervised learning differ from those of, say, ResNet. What different properties do they capture, and how do we characterize that in the process of learning so that we can build more robust learning algorithms? And finally, we'd like to release open source code and methods to the research community to continue to study some of these properties. These aims, at the end of the day, the goal of both my lab and a lot of the other researchers working in this space is to move from correlational to think about how we can make causal predictions using some of the data that we have. And my students and I are thinking of a lot of problems and how to make use of causal graphs for problems like density estimation and leveraging deep generative models, which are a class of Bayesian networks, as flexible structural equation models to answer counterfactual queries. We've been thinking about questions of what are the challenges when you deploy machine learning. So we have students working on new algorithms for the identification and detection of covariate shift, and a student working on thinking about how to reimagine risk score discovery with new machine learning methods. And so if you think of risk scores such as the Framingham score and the MELT sodium score, they're ubiquitous in the clinical landscape. And so we've been working with Mamta Bhatt, a clinician at UHN, to think about how we can use ideas from machine learning to build data-driven risk scores. And so I think that brings me about to time. So thank you all very much for your attention. I'm going to pause now and take any questions that you have.
Thank you so much, Rahul. That was really an informative talk, and we all enjoyed it. Thank you for the time. So we have it's time for questions. If you have any question, kindly put that in the chat. I'll relay that to Rahul while he answers the question. And then I have one question here from Hadi Mohadas. Rahul says that the interventions can change the dynamics of disease development, and the response to treatment could vary over the different subgroup of patients. However, in practice, the margins are not clear all the time, even for the clinicians. How could the models overcome this challenge?
Yeah, that's a really good question. And so, that is a limitation of the model in AIM-1 that I presented as it stands. And you're absolutely right that you could have heterogeneity among subgroups in how the interventions manifest and the effects that they have on mitigating the disease over time. And so I have a technical answer to you, which is you would incorporate the idea that there are subgroups as an assumption into your learning model and modify the learning model, which is a really cool idea for future work. that we have students working on, but it's not something that I talked about right now. And that, that method will, to some degree, mitigate the concern that you have. But I should say that that comes with a caveat, which is that if we want to also capture this underlying cluster structure in the data, then we need more patients. And so it's not, it doesn't come for free. And so the second question, sorry, I can see the chat right now. Hopefully that answers your question, Hadi. If not, please do ask a clarification. The second question is that two approaches being used in digital histopathology versus smart search. Latter is something like a recommendation system. How do you compare the pros and cons of these two approaches? Right. The idea of a smart search is, I think, so the two approaches are very distinct and serve very different populations and different people within the clinical system. And so a clinician might be interested in a smart search if at the time of diagnosis, they want to get a sense of who are the other patients who have similar clinical manifestation to this individual, and what do they look like and what their trajectories look like into the future. So, but that same clinician might also have an interest perhaps when grading the cancer to know what a model's assessment is. So I can foresee that I can foresee two different, both of them being very useful, but I would say that they're used at different stages of the clinical pipeline. In terms of pros and cons, I would say that the search, the smart search, which operates something like a recommender system, has, in some sense, you just need to output a good ranking. And you can just rank all of the other patients in the hospital database based on some notion of similarity. And you can let the doctor decide whether your ranking is good or not. In the context, I would say that the bar for doing smart predictions has to be much, much higher. We have to think harder about whether or not these predictions are calibrated across different subpopulations, whether they're fair, whether these classifiers have any inherent bias that we need to worry about. Because whenever you make predictions, it is a quantitative assessment that is being prescribed by a model, and we don't yet have a good understanding of how doctors would make use of that information in clinical practice. So I'd say that the bar for a smart predictive device should be higher than something like a smart search. Nigel asks, you mentioned covariate shift as the current ongoing work. Are you trying to compute the complete covariance matrix calculations for each data set? Not exactly. Sorry, I don't think I did a very good job of explaining what covariate shift is. Covariate shift is essentially this phenomena that you deploy a classifier on a data set. Let's say you build a risk stratification tool and you deploy it in a hospital. And then 1 year later, COVID hits, and suddenly your classifier just stops working altogether. And if we think about why, it's because the underlying populations that are now being applied to this classifier look very different from the populations that we would train the model on. And really, this goes down to the idea that in machine learning, there's almost always an implicit assumption that our test distributions are drawn from the same, our test data, all the data that we see during deployment are drawn from the same distributions as the data that we train the model on, which is violated in the case whenever we have covariate shift. And so hopefully that clarifies some of the distinctions between covariate shift and why we need technical solutions to this problem.
Okay, thank you so much, Rahul, for the answer to those questions. While we wait for further questions, I just want to know if it's okay to leave your maybe contact on the chat, if possible, for someone to reach out to you for more questions, maybe your LinkedIn connection, or maybe your email, if possible. Okay, thank you so much. So in case if you want to maybe clarify, if you want more clarification from Rahul about the talk or want more information, you can reach him through the email he provided here, so or maybe through a LinkedIn connection. Okay, just a reminder to visit our booths to take advantage of the offers we have from our sponsors for this event. And then if you missed any event at all, you can look at the replay button to play some events that you missed, some of the talks that you missed during this conference. So thank you all for taking our time for this talk. Thank you, Rahul, for the effort and the time you spent in preparing the talk and all that. So hopefully we'll get to see more during the rest of the conference. So enjoy your conference, everyone.
Thanks, everyone.
Okay, bye.